I've given you a brief idea. I think you have now a brief overview. You might not understand it fully. It's perfectly fine. Even I don't understand it fully. And I might say that is true for even those who are working for 10 years. So it's quite a broad subject, and I love this one. I was discussing with a friend, and we thought that what is mass spectrometry based proteomics? It's the use of physics to identify the chemistry behind biology. So it's quite varied, and beyond physics, now we have Professor Graham who will tell how to use mathematics to make sense of it. So it's quite interdisciplinary. You just dived into mathematics. But today we will be stating that how do we take a sample that is, I don't think I need the mic. If I'm not audible, do let me know. So how do we take a biological sample that is very complex, non-linear, as Professor Graham was telling, and put it into an instrument that does not understand much of biology. So we have to prepare the sample likewise, and as with all of us, the samples too are very diverse, and it can come from diverse sources. So what are the samples primarily that we can put into a mass spec? Actually, we can put into the mass spec anything. We can put in solid tissues, samples from solid tissues, not of course a solid tissue. Don't do that. It would be ruining about 10 crores in 1 second. Cell lines, serum, plasma, swab, saliva, urine, and fecal matter, plants, fruits, anything that is a biological sample can be used to make sense. And how do we do that? I mean, if I have to give you a very brief overview, we take different types of sample, we extract the protein, we do trypsin digestion, it's also called tryptic. Not always with trypsin, we'll come to know. Then we desalt the peptides, and then we finally put it into the mass spec. Right? This is a very generalized overview. Now we'll get into the specifics. And before we do that, we have to do a SDS page check for QC. I hope all of you understand what is an SDS page. Sodium dodecyl sulfate polyacrylamide gel electrophoresis. It tells us about the sample integrity. Yesterday, Professor Graham was telling that integrity of the sample is very important when you are doing any kind of AI/ML based study. You see, anything that is extrapolating your data needs to be clean, and your source of data should be clean. So we do an SDS page QC check, and what we primarily check for is whether the sample has degraded or not, right? So anybody who has not seen an SDS page gel ever, all of you have seen. And what do we see there? We see bands, very sharp bands. But what happens if you don't see a band and we see a huge smear? It means your proteins have degraded. Now you will later understand. And what do we do with trypsin? We break it down, the proteins. So isn't it very strange that first I'm telling you your protein should not be degraded, and then I'm telling you degrade your proteins with trypsin? Right, I'm not kidding, but that's what we do. This is for bottoms-up proteomics. There is a top-down proteomics which is not part of the syllabus. So why do we do that? You see, it's like in bottoms-up proteomics, what we want to do is we want to recreate a broken vase, a flower vase. Now, suppose you have a very small camera or a microscope, and you want to. Have you ever done a panorama shoot with your mobile phone? So why do we use a panorama? Because the field of view of the optical sensor is not that broad. It stitches, right? So a mass spec cannot identify the entire work for your alternative protease. So whenever we work with an alternative protease, again, lots of standardizations to be done. Sometimes people use something known as the multi-protease strategies, where you don't use one, but you use two. So you have the good part of trypsin, and then to avoid the bad parts of trypsin, suppose it has generated too many long fragments, you use another peptide in conjunction. So what it does is it improves sensitivity and accuracy of protein quantification, expands the sequence coverage, it can identify unique peptides, it facilitates the de novo assembly, and one of the biggest advantages, it can also help you in better PTM or post-translational modification identifications, and of course increased protein diversity. So usually that is what has been observed is when you use multiple proteases, you have a better proteome coverage. But in most cases, trypsin is good. So once you have digested these proteolyzed, these protein, you have peptides. Now we'll stop using the word protein. Now what we have in our mixture are peptides, and these peptides need to be purified. In the very first step, if you remember, I showed you the general schematic. So these peptides need to be purified, and these purifications are done by specific tools. So what we need to do is mostly it's solid phase extraction columns. There are other specialized techniques like stage tips, SP3 and S-trap. So S-trap is especially useful when you're using some specialty buffers. Again, these are technologies that has been primarily patented. Solid phase extraction columns are the most used type of methods. So what we do here is we do a column conditioning, right? So this is for the waters. I think it is visible to you, those at the back. Right. So we do the column conditioning with 1.1% TFA and 90% methanol, and then we equilibrate the column with 0.1% TFA in water, and then we put all our digested peptides into that setback column. What happens is the column will bind with the peptides, right? It will bind with the longer peptides, it will bind with even the peptides that are shorter, and of course those of ideal length, like between 7 and 35. Then you pass 0.1% TFA and 5% methanol. What this solution does is it creates, so 0.5% methanol is in water, right? What it will do is it will take away all the salts and buffers. So this elution, with this desalting, it will create a lot of elutant, and that you have to discard because that is primarily salt. All your peptides are very strongly bound with the C18 material that is in these solid phase extraction columns. Once the C18 material, these salts have been moved away, we dry this column. Then we put 0.1% TFA and 50% acetonitrile. What it does now is all the peptides that were now very tightly bound will start to loosen up and will come out in the elutant. Once it has eluted out, all we need to do is vacuum it, and the acetonitrile will evaporate, and even the ABC buffer, all the remnants, they will all evaporate out. Urea is already out. Now you have clean peptides. A good way of understanding that the purification has been done and it has been done well is you won't be able to distinguish a microphage tube that has peptide with the one that does not have a peptide. But if you start getting yellow spot or maybe faint white spots, that means you have done something wrong somewhere, and peptides are usually colorless. You don't see them. So I think I have already explained, so I will not go through it, but I will share these materials. So what we use, and these SPE materials, the solid phase extraction materials, they come in various shapes and sizes. So you have micro columns, you have cartridges, you have plates, you have micro pipette tips, you have magnetic beads, depending on the type of work you want to do. Suppose you are working on thousands of samples, it won't be possible for you to do it individually. There you will go for this plate format, a 96 well plate format, right? and you will have liquid handlers and robotics to assist you. So big labs that process lots of clinical samples to do their studies, they use automated liquid handlers and plate format desalting methods by which they can get very clean peptides without any issues, and it's completely automated. And the mostly widely used material is reverse phase, and it is used for protein peptide fraction. You have ion exchange columns, but it is rarely used. You also have Heilig columns, and this is preferred when you are working with glycosylate proteins and peptides. So again, you see, if you are working on a post-translational modification, you cannot use a reverse phase column. You have to use a Heilig column, normal phase, right? So fact of the matter, I think I have been able to, and I'm repeating it time and again, it's not one size fits all. Your experiment, your biology will determine what tool you will use. Tools are many, right? Okay, so I think I missed one part. Get back to it. Okay, so sorry for this. So while we were talking of digestion, I just missed one part. Was before you go for digestion, you have to go for reduction and alkylation. So the point is when you are going to digest with trypsin. So go back to that slide when we were discussing about trypsin. So when we have to digest with trypsin, one of the big challenges is proteins are in its normal form, not in a straight line, right? They're all jumbled up, and the reasons of having these native conformations even after cheotropes are these secondary structures that it forms due to disulfide bonds. Now you have to break these bonds, and these bonds are very persistent. So once you break it, there is a tendency of these bonds to scramble back, not at their exact positions, but maybe at some other position. But anyway, your proteins will fold, and what happens is it inhibits the proteolysis. To enhance your proteolysis, what you can do is you can do a step that is known as reduction and alkylation. The first step is reduction. One very good, most commonly used is TCEP. So you can use TCEP. It's there is diethioethanol or DTT. We also have beta mercaptoethanol. Mostly used sample is TCEP, right? Now once you have broken the disulfide bonds will most likely again scramble back. To prevent the scrambling, you have to go for an alkylation steps. So what they do is they cap the free thiols that you have just generated, and it prevents the reformation of disulfide bonds. And one of the many reagents that you can use is iodoacetamide, is the most commonly used that we use, iodoacetamide. Also you can use for alternative as N-ethyl malamide. So these are again alternative tools. Alternative tools are there when you have a specific reason of not using iodoacetamide or chloroacetamide. So this is what happens is this. So you have folded aces bonds, you add DTT or TCEP, the bonds are now unfolded, but it's still these thiol groups, and they have this strong tendency to fold back. Now to avoid, you have to cap these thiols, and you add an alkylating agents. So these alkylating agents will cap the thiols. Now these thiols can no longer scramble back and form the native protein. You add trypsin, you get better cut sites. Okay, so we've gone through a lot. I think we are still have 25 more slides, so maybe we'll try to cover it up by 11:15. Then I will hand it over to Professor Graham. So once you are done with the protein, so remember we had started off by measuring the total amount of protein, right? We did Bradford or BCA fluorogenic, again based on the needs of our experiment. And now we have digested it, and we have done a peptide cleanup, And now we have purified peptides. Now these peptides also need to be measured. If you remembered yesterday when Professor Sanjeeva was showing that his new timsTOF can work with 100 nanogram peptides. So you have to quantitate the peptides, 500 nanogram peptides or 1 microgram peptides. So without quantitation of peptides, you cannot just inject a lot of peptides into your LC. It will give false results. So you have to do assay for that, and you cannot use assays like BCA to estimate peptides. The best ways are, first you can use fluoroscanning based assays. So these are again fluorescence based method. You can also use tryptophan fluorescence. That most of us we do that. We use something known as the SCOPES method. You can look up SCOPES, SCOPE apostrophe S. So we use SCOPES method where we measure fluorescence of tryptophan using a traditional UV visible spectrophotometer, but of course in a microplate format. Now you won't use a cuvette because you did not purify that much of peptides. So most SP columns that we use in our labs get us around 20 to 25 microgram of peptides, and once you have done that 20 to 25 microgram of peptides, you don't want to lose it, because that peptide will be run on a mass spec. First, if you're planning to do some discovery proteomics, once you are done with the discovery, you would again want to use that peptide to do some targeted studies. So you don't throw the peptides just like that. So extractions are usually for one sample type, you usually do it once and take maybe 2 aliquots, keep it in the fridge. So what we do is, at 205 nanometer it measures the peptide bonds. At 280 nanometer it gives a strong fluorescence due to tryptophan and tyrosine residues. So this is a microdrop plate, and this is the instrument Multiscan Go. This is from Thermo, but of course you can use in your own lab. You can use almost similar principles, and you get the ratio of 280 by 205. It gives you also a good measure of digested peptide. So if you find a strong amount of peptide bonds in your sometimes you can pick up just by a ratio that some of your protein did not get digested properly, or it passed through the column. So these kind of stuff can be picked up. So this is a very basic method. I've spoken for almost 1.5 hours. I have 30 more minutes. So we'll discuss. We're running out of time. We're already running 30 minutes late. So write it down. So plant proteomics, and I think a few of you yesterday told me that you would like to see how we use plant proteomics. So plant proteomics comes with unique sets of challenges, which we'll discuss first. So first unique challenge is it's a very strong cell. It has a cell wall. It does not have a cell membrane. So the first thing is, if you are trying to extract proteins from plants, you need to have a very strong homogenizer. So vigorous homogenization, especially like grinding, bead beating mills, and rotostator homogenizers, you need that part. Second, presence of large vacuoles. Now vacuoles are present in cells, and they can contain substances that can degrade or denature the protein. But when you will lyse your cells, you will have the problem, because now you're also lysing the vacuoles. Second is lots of phenolic compounds. Most of the plants will generate lots of plant-specific metabolites, and all these will interfere in the process. So what we do in protein extraction for plants is we rupture, and when we start mixing, we need specialized methods for isolating proteins from organelles or subcellular fractions. So we have to be very careful. And again, the type of plant that you're using, the kind of family. I'm not from botany. The last time I studied botany was in my undergraduation days. So the family of the plant, the kind of metabolites, and also the plant part. Are you working on a fruit, or a vegetable, or a leaf, or a root, or a stem? All these things will, and even for a leaf, what kind of leaf are you working on? The one that is green, the one that is yellow, the fresh leaf, or the dried leaf? So everything will determine the kind of protein content that you can derive from plants. Next is low protein content. That's always true. So compared to other organisms, plants have smaller fraction of cytoplasm. So if you remember the plant cell, we have a huge vacuum. The cytoplasm is minimal, and this vacuole will contain all the stuff that you do not need, right? So there are a few strategies that you can use. First is to prevent protein loss and degradation. You can use plant-specific protease inhibitors. Also, you can address the phenolic oxidation with buffers, with reductants, and specialized additives. So you need to know the chemistry of the plant that you're working on. Based on that, you have to add buffers. Best part is look up on the research articles that people have done, and if you have any specific books that deal with plant proteomics, this is not what most people do. So you have to look up specialty areas. Of course, you have to work with low temperature. So low temperature is something that is true, that holds for animals as well. And of course, we have plant-specific protein denaturation. You have to use protein denaturation to prevent and to stabilize the proteins and prevent degradation. So these are common methods. And also, you have to remove the interfering agents by using precipitation strategies, right? So at multiple levels, you have to use some techniques. So what all we can do TCA precipitation or trichloroacetic acid precipitation. You can use ammonium acetate precipitation. You can use acetone, and all these, what they will do is primarily precipitate out the proteins, and then once you are done with the protein pellets, you have found the pellets, you can again use methods, chemicals to resolubilize and the detergents, the proteins. So you can use detergents, you can use phenol, you can use chemotropic agents like urea to again dissolve the precipitates. But what it basically does is prevents your proteins from getting impacted by the phenolic compounds or any other metabolite that the plant is storing, right? So if you want, a good example would be if you want to understand the proteomics of a ripening mango. So imagine a mango when it is green. It has a different flavor, it has a different texture. Again, when it is ripe, it has a different flavor, different texture. So you cannot use the same protocol for the ripe mango that you are using for the green mango, right? The mango that is just very unripe mango. So please be very careful when working with these samples. And of course, since you are using two different technologies to extract, you might be using two different technologies maybe to extract, especially for stuff like fruits or maybe for leaves. So you are wanting to understand why does a leaf get brown during the spring. So you want to understand the proteome profile of that. You cannot, if you are not using the same methods of protein extraction, you are adding one more variable. That becomes a limitation of your experiment, right? But that's fine, rather than not doing the experiment at all. So, of course, just like we discussed, we have to think that the extraction protocols should not influence the downstream results. So you can do so like one method that people usually try to follow is, sometimes somebody would have done a 2D gel electrophoresis study some 15 years back, and you want to do LCMS based study, and you will want to use the same protocol. Don't do that. Those are two different technologies, although both are technologies that are specific for proteomics, but these technologies are different. You don't use something that is developed for 2D for LCMS. So you have to adapt to the shotgun proteomics approaches. There are sample preparation techniques that you're usually using, like this, as I told, FASP, I was talking some time back. So you have filter-aided sample preparation. It minimizes the contamination. There is also some specific techniques like the SP3 technique, the single pot solid phase enhanced sample prep. So they use coated magnetic beads for efficient preparation. So basically there are magnetic beads that are coated with SPE material, right? And then those SPE material will preferentially bind with the peptides, and then all you need to do is a magnetic separator, separate out the SPE beads. Now you have very efficiently removed anything but the peptides from the sample, right? So it gives you higher throughput, it gives you higher reproducibility, but of course what is not mentioned here is it also gives you a higher bill, something that you need to think of when you're designing your experiment. Another challenge is hyperabundant proteins. So this is a challenge that we often face. So in plants that are specifically the green tissues of C3 plants, they have rubisco, right? I'm not from botany, but what is rubisco? It's an enzyme, and it's hyperabundant. And just like we were discussing that we should not have too many abundant proteins, then they will mask your results because it won't allow you to see the finer details, the finer structure. So also there are specific strategies to target rubisco and C storage proteins to reduce the dominance. So you have to think about it, that when I am designing an experiment, like if it is a green tissue of a C3 plant, what might be the challenges? Has anybody worked on a green mango, right? Or a green tomato and a red tomato? So how does the tomato change colors? But again, not only does it change the color, it changes the shape, the texture, right? So you have to keep all these things in mind when you're designing your experiment, and you have to read a lot. And don't believe too much of what is there on the internet, except if it is not from a peer-reviewed journal paper. So what are the challenges? Just a small recap. First is cell wall, large vacuole, interfering substances. Next, low protein. So you don't have too much of protein to work with, and sometimes you have too much of one protein, that is rubisco. Effective strategies can be binding, breed mills, plant-specific protease inhibitors, and advanced precipitation techniques like TCA, ammonium acetate, acetone. You can also use sp3 and FASP, and also use those magnetic beads to separate out your protein. Next is the blood plasma proteomics, almost same as plants, but they have their own, so again you have some specific challenges here. What could be I think I discussed initially. So what is the challenge for blood? Exactly, it's very rich in protein content, so you will have issues. So the protein abundant protein mass. So 99% of the total protein mass in human blood is primarily, human blood plasma is primarily just the top 14 proteins. What we do? We deplete them. So how do we do it? So we have to deplete or remove them. Just like rubisco, here rubisco was 50, they are like 99. So first, without depletion, it's very hard to get anything, any information that you can gain from blood plasma. And blood plasma is very important because anytime you want to find a biomarker using mass spec, people will ask you, can you find me a blood-based biomarker? The reason is, it's very standardized setting. If you can find a protein that can tell that, okay, this person is suffering from this disease, or maybe this suffering, or like the work that we did at IIT Bombay was about whether this person will have severe COVID or the COVID will be of non-severe types. But this is a clinician who will come to you and ask you very direct questions. They won't spend much time understanding how the mass spec works. And so as a scientist, you have to take care of it, how the mass spec works, and give him 1 or 2 proteins, or maybe a panel of 3 proteins, right? That can effectively prognose a disease or diagnose a disease. So how do we remove these top 14 proteins? There are methods like the dye-ligand depletion method, where you target the serum albumin by a dye called the Cibacron blue. It has electrostatic forces and hydrogen bonding and hydrophobic interactions. But the biggest advantage, it's low cost, it's robust, it has a very strong binding capacity, but it comes with a big challenge it lacks specificity. So sometimes in one batch of the dye will bind, the next batch might not bind. Or even within the batch, there will be batch to batch variation. And when you induce variation, you stop seeing the biology. You start seeing artifacts. You have induced variations on your own sample prep. What you're seeing as differentially expressed proteome or the DEPs, and now you're making heat maps, you're making models and telling this is my panel, and what is your panel showing is, okay, this was the difference that you got while processing the samples over the past 6 months. That's what your sample is showing. It's not showing a biomarker as such. Also, you have specific protein ligand depletion columns that act via interactions from bacterial cell walls. There is protein AG or protein AG from bacterial cell walls, but again, they are non-specific binding. So non-specificity is something that we want to avoid. To avoid that, we use immunodepletion, and immunodepletion works with antigen-antibody interactions. So you have antibodies against the top 14 abundant proteins in plasma or serum. When you put the serum and allow it to incubate in that slurry, what happens, all the antibodies will go and bind with their antigen, that is the top 14 proteins in the blood. And then all you need to do is separate this antigen antibody complex. What you're left with is the entire blood proteome except those top 14 proteins. It's very highly specific, so there are commercial kits for that. So Agilent gives the mass column, Thermo Fisher gives high select beads. There is also something from Pierce, which is again a brand now owned by Thermo Fisher, but as I said, it's extremely costly, around 14 to 15,000 per column. So if you have to process maybe 100 samples, at least to get something of big data to give to Professor Graham, we have some data at least from 100 patients. That itself will cost you in lakhs, in crores rather. How much is 14,000 multiplied by 100? 14 lakhs. Then add all the other steps. So each sample for blood plasma in India, at least in 2021-22, it would cost around 35 to 40,000 one sample. That's the cost of processing one sample from plasma to the proteome chromatograph. Also, you have to consider sometimes when you're washing it, you might induce artifacts, right? Sometimes you can use a combinatorial peptide ligand library. It partially depletes major proteins, and it enriches when you deplete the major proteins, and what you have is you have an array of ligands. There are 6 amino acid peptides. It normalizes the globin protein abundance, but again a very big drawback is non-specific binding. Loss of proteins during elution, inefficient binding. So these are all challenges. Other methods can be precipitation methods. So you can use organic salts, inorganic salts, organic solvents, non-ionic polymers, reducing agents. So you have multiple methods to work in plasma. Precipitation method is one. So the paper I was talking about from Professor Hanostein's lab, they had used TCA to precipitate because they were working more than 1,000 samples. So they had used TCA. Fact of the matter is, if you are to find the perfect method, you won't find any perfect method. Every method will have its limitation, right? That limitation might be money, that limitation might be a lack of sensitivity, but whatever you follow, please follow one method. Don't jump from one method to another method, at least in one practical experiment. Don't do depletion columns, top 14. So you had money. For the first 15, you used depletion columns. And suddenly you got hit by poverty and you moved to precipitation methods. 20 you did with precipitation. Then you realize that, okay, I'm not getting good coverage, let's do this. Now in that 100 samples you have used 6 different methods of protein separation, depletion of major abundant proteins. That way it won't work. So think ahead, plan your experiment well, all the upstream and downstream factors, and then decide upon the methods. There are emerging technologies that use bacterial cellulose, nanofibers, cryogels, nanomaterials. So there is something known as the nanoparticle protein corona workflows. They allow identification of around 6,000 proteins in plasma and serum. So primarily protein coronas and extracellular vehicle EV vesicle enrichment techniques. So these are newer technologies. So you might not have access to them. So be careful when you're planning anything. Look for access. Like in our own lab, we can tell that we wanted to use Protify in my lab at BHU. So we could not because of some challenges in the supply chain. Okay? So this is in summary about blood protein depletion. So I think I have some time. And volunteers, do you have tea? So Professor Graham, done this? Okay. So I will stop here and I can take a few questions. If you had some question. Anyone? Wait. Recording on and? Right. Nanometers?
2.5 and 280.
Right. Yeah, that's the Scopes method.
Yeah.
That's the Scopes method.
So like what range and what is there a ratio which gives us the purity and like what is the?
So usually we have a ratio. I can show you the table. But that will give you a proper understanding. So we calculate the extension coefficient ratio and we try to keep it less than 2. More than 2 and between 2 and 5 actually. So beyond that it becomes very difficult to work on the mass spec. So and every lab will have its own method. Few people actually the fluorescence based methods are much better, much. But again they are kit based and they are patented, so adds on to the cost of sample prep. Also there are newer tools like the timsTOF that Professor Sanjeeva was showing the other day. They have this quantitation method that is already integrated in their workflow, like your mass spec does it. You don't have to do it. So they have column based SPE cartridges. These small columns are put in small micro tip boxes. It's a completely automated workflow. So sometimes you need to have some level of automation. Sometimes you can do if you have lesser samples. So you see, the lesser the sample number, the more human interference. The moment the sample number goes beyond a specific set, maybe you have to process 100 samples a day. That is not humanly possible. And it would be humanly possible if 10 people started working together, but that will add individual variation, experimental variation to the data. In the long run, it's very difficult to work with a data that has so much of interference just because 50-60 people process the sample together. That's one of the challenges. Anyone else? Yeah, please.
Sir, there are some samples like chlorella, like green alga or plants where we have thick cell walls. We give a long intervals of ultrasonication. So will it also degrade the protein content?
Ultrasonication long intervals?
We have
how long?
Duration like 15 minutes, 5 minutes, with on and off.
Okay, like how long? Like amplitude, like how is the pulses? How long pulse? Each pulse is like
10-second pulse on and 5-second pulse off. That is how we do it, and we do it for 15 minutes because other than that the cell wall won't break for nice patients.
Right. And you do it on ice?
Yes, I do it on ice.
Ideally it won't break. And what buffer are you using? Urea?
No, Chris.
Tris. Okay. See, as I said, there are no foolproof methods. You can use some enzymes to degrade it as well, but the moment you add enzymes, you add noise. Right? So you have to standardize. I mean, since I do not work on cell and plant cells much, so I cannot give you the exact methods. But there are groups in NIPGR. I think Professor Shubhra Chakraborty. She does plant proteomics. NIPGR, that is National Institute of Plant Genomic Resources. So Professor Shubhra does. There are a few other people who do plant proteomics. You can contact them, get in touch with them, with their lab, and also go through their papers if somebody is working. And it is taken from paper, or do you have standardized it in-house?
Actually, the duration of ultrasonication, we standardize it in our lab itself.
And how you are standardizing it? Like,
During the quantitation, like we quantify.
are you running SDS gels every time?
No, not every time.
At least when you standardized it, did you run an SDS gel to see how was the protein quality?
Yes, we did it. The bands were visible, but there was a stream too. So it wasn't that proper bands were visible.
There was
There was a stream, like it wasn't proper bands were visible.
like a smear was there.
There's no
Proper bands were not visible.
Not proper.
Not proper. So it was not bad.
Yeah, it was not bad, but it wasn't good.
It wasn't good. So you gave me the answer. So maybe if you have time, you can try a few more methods. You can increase the pulse, the rest pulse, right? 10 seconds. Maybe induce a 15 or 20-second gap, and increase it to 30 minutes. See, the point is ultrasonicate probes. Is you are using a probe sonicator. Probe sonicators tend to heat up a lot, right? And what's the amplitude that you're using?
50%.
So 50% is on the higher side. Usually we use 30 to 40. So, but that's for animal cells. But the good way is to avoid heating. So as we discussed, you can, please sit down. So the good way is you can do some QC checks on the gel itself. Seeing is believing. Other day I think Professor Rizvi was telling. So if you can see proper bands, clean bands, then you know that. And as we have discussed that if you get lots of smears, what we are effectively doing is you are killing off you are chopping off the proteins even before it reaches the mass spec, and it will create lots of noise. So have you done a mass spec yet? No. You're in the first year of PhD? Second. So you have some time. Maybe for the next 2-3 months, all you can do is check what a 30-minute pulse would look like. I mean, 30-minute ultrasonication would look like with 10 seconds of sonication, a 20 seconds pause, and again a 10 seconds. So you can do this. So every minute you give 2 10-second pulses, or you can use 5-second pulse, 10-second gap, again a 5-second pulse, 10-second gap. Even then you would effectively reach the same amount of pulsing. And use chilled ice. I mean, put it on ice, keep it chilled, because the problem is the microenvironment will heat up. You can also now there are so many buffers. I will give you a few papers to work with. Buffers you can change, you can add or alter the buffers, you can add plant-specific protease inhibitors. First thing, the very first slide that I showed, you should have clean bands. If the bands are not clean, even if you run it in a mass spec, whatever you would be getting is probably noise, it's not the data. We can discuss more. Anyone else? Yeah. Anyway, can some volunteer pass on the mic? Maybe. Yeah, please.
Sir, I tried.
I'm sorry, please tell your name, because anyway I will forget, but still.
My name is Gina.
Jimal. You are? Simran, right.
I'm from Naika, Ahmedabad.
Right.
So currently I wanted to ask regarding that I have used molecular weight cutoff filters for like the biomarkers that I want to extract out of the plasma samples are of low molecular weight. So ideally what molecular weight cutoff filters are used for is we take the part which is like the parts which remains above the filter. What should I say?
So you have, let me know, unless if I have understood you right. You have used molecular weight cutoff filters.
I
So
took the filtrate and then I processed the filtrate because I want the low molecular weight proteins.
low molecular weight proteins.
But then also I'm not able to detect it in your mass spec. After like.
So this is plasma you are talking about.
Yeah.
Rat, human, like,
Rat plasma.
so you can kill many more rats if you have ethical permissions. With humans, it's very difficult. So why don't you, and this is from some paper that you have used, or
No, like
in-house?
I'm doing a trial and error method.
So see, abundant proteins and molecular weight proteins, I don't know how well they correlate. So you need to tell me when you filter, the filter that you're using.
So basically what I'm trying to do is the albumin or the immunoglobulins, they are
Well,
having higher molecular weight.
right. And when you're using this filter, what are the conditions of using this filter? Like, are you using any buffers along with that?
Yes.
What?
PBS.
PBS. So you're diluting your sample
Yes.
with PBS,
Yes.
chilled PBS,
Yes.
and then you're passing it through a filter, a molecular weight filter.
Like, but the manual says that the user guide says that whatever remains on above the filter, we have to use that. So people use it for taking the high molecular weight proteins, but I am using it in the reverse way. Is it the factor is affecting my?
So what does your mass spec say? Nothing, or
It is showing nothing, like no proteins are there.
okay? Before you put it in the mass spec, have you ever checked in the filtrate what is the protein concentration, like using just bad food?
Yes, I perform BCS. So that is giving me a good protein. Globulin is quite high. And after that, when I digest the protein, and then when I'm really mass spec, I'm not like able to detect anything.
Anything like,
Nothing is
how does the chromatogram look? A straight line? No peaks?
just noise, similar to the blank sample that I'm using.
Okay, so a few things first. I'm not very sure about molecular weight cutoff filters for plasma. I mean, if you have read it somewhere, I don't know. I did not use it ever, because what we used molecular weight cutoff filters were for doing FASP, this filter aided separation techniques. So there we used molecular weight cutoff filters to remove these contaminants. Also people use molecular weight cutoff filters to when we do some metaproteomics, like for VTM, for sewage water, all those kinds of stuff. Since I don't know much, I would have to look into your details before I comment anything on a public platform. So maybe we can discuss sometime about your specific problem. But since your mass spec, you're telling me your BCA assay is showing results. Have you tried some other assays? One point could be that whatever you're getting in BCA is noise. It might be the filtrate that is coming out somehow gives some results, like these are all chemicals, right? They might react. So have you tried running that filtrate on a SDS gel?
Yes.
Is it showing bands?
Yeah, it is showing proper bands.
So you have protein that you can detect, and somewhere after digestion.
Like after digestion, maybe something is going wrong within the digestion stage.
So what kind of desalting are you doing?
Pure peptide desalting.
And what kind of, you are taking all the gradients separately, like 30, 40, 50, 80% acetonitrile. So you are pooling it all together, or you are taking separate gradients? When you are eluting it, so you are, I showed here 50% acetonitrile. That's a generic way of doing it. But sometimes people use different desalting methods. So they will take a 20% acetonitrile, then they will go till 80% acetonitrile, because some peptides will still stick on at 50% acetonitrile. For that you need 80%. And also the gradient that you're running on your LC, the acetonitrile gradient. What's the gradient? A lot of factors will impact, right? So the gradient sometimes, is it a short gradient? Like how long is the run?
120 minutes.
A 120 minute
And
nano LC, like what's the instrument? Q Exactive? Orbitra?
yes, orbit.
Which method? Q exactive or fusion Lumos? Fusion. 120-minute gradient, and in all probability it will have a 30 to 80% curve, like
Yes.
the elution profile. Seriously, I don't have an answer right now because I didn't look at the raw data, but there are multiple factors that might go wrong. So since you're getting, so see, estimations like the BCA and all these things, it can fool you. So try doing, since you're using low molecular weight proteins, what kind of staining are you doing?
For
CBB?
For SPSP,
Right.
I have tried for Masi also silver staining.
Okay, so you have done all. So in silver staining, does it give, can you see on the lower molecular weight, can you see stuff?
Lower molecule, yeah, I am getting
Clean glands.
yes, yes.
A good way of doing it now would be take those bands
And perform in gel.
and perform an in-gel digestion.
But again, the problem is because I am using the nano acid, I might not be allowed to, because salt content might remain the acrylamide.
I don't think in in-gel, you do it digestion in in-gel, but then after the peptide purification, you are not sending the gel into the mass spec, you are sending just the peptides. So, yeah, so you are in the second year of PhD, like third year.
Started off with it.
Third year. So those who are not into PhD, so you will come up with these problems that would require extreme custom issues. I mean, and hats off, she has done almost everything that's in the book. Now, Gina, right?
Also tried there's a paper for actually I am trying to separate out 3 biomarkers. I have found a specific paper for intact analysis of one of the biomarkers, and I tried protein precipitation from that paper exactly. Then also I was not able to detect any of them.
So you are using this facility at NIPO.
Yes.
I'm not advertising, but Professor Sanjeeva's lab has really good people around, right? And Professor Sanjeeva is really informative about this. So why don't you contact him? You can take reference that if we met at the Gyan course at Allahabad and I had these issues, you can meet him, or Ahmedabad to Bombay is not that far, or you can write him an email. And sometimes he does this internship training programs. You can land up in his lab. He has a new instrument. So sometimes it might be an instrument issue as well. Like the instrument is failing to, the column issue, like you are not using your own column, right? They have the facility column.
C18 columns.
And a C18 column, but it might be that the column is failing to pick up those peptides. Sometimes the columns age and they don't give much good results. So there are so many variables along the way. The buffers you're using, the solvents that you're using, the column that is being used. Now how much of blanks do they run? How much of the column was storage? Sometimes few facilities which run all throughout the year. They usually perform better than facilities who did not keep the column conditioned and it was kept out in the dry, and the column dries up, the C18 material gets wasted, and then we will have these issues. And always ask the facility how much is the HeLa coverage they are getting, or yeast digest. So they would have these information. So that will also help you understand whether, since you have already removed a lot of the proteins, all I was expecting is that you will get a spike. 1 or 2 spikes at the lower on one of the side. Also the proteins, what hydrophobicity does it have? So you're using trypsin, right?
Yes.
So the multiple things you can do. Maybe you can add maybe the protein that you're looking at. The trypsin digestion is not giving much of variation, right? I mean, we just went through that. The peptides cannot be too long, it cannot be too short. You have to hit that sweet spot. Maybe it's not hitting the sweet spot. And the peptides that you're having are not getting that flight. You can add one more enzyme to it, lyse C to it, or many ways But since you have already done lots of troubleshooting, I think you can ask your supervisor. He or she can get in touch with, again you can get in touch, but of course do take permission of your PhD supervisor before getting in touch with another scientist, and ask him. So he might be in a much better position, and maybe he can also offer to run your samples to do some troubleshooting. I don't have a mass spec here, so I can't comment on that. But kudos to you. You have done lots of troubleshooting all by yourself, and that's why PhD, despite being a 100-pager or a 200-pager, takes 5 years.
Can
Yes.
I add?
Yeah, sure.
So I also work on serum plasma proteomics. You can discuss with me. So during your discussion, I have some thought. You can
Yeah, sure. Yeah. That's why we are here. We are all collaborators at stage. Anyone else? So if you don't have any questions, I will thank myself, being the coordinator, and then I will invite Professor Graham for his talk. So after Professor Graham's speech talk, we will go for lunch. So he has 2 talks today. One part is yesterday's talk, and maybe the lunch will get delayed. So maybe by 1:30 we will hit the lunch, and after 2:30 onwards we will discuss on few mass spec more things. And if you have any specific questions, you can also drop an email to me, which I would try to respond after 30th. Till 30th, it's pretty busy. Okay, thank you all. So now I will invite Professor Graham for today's talk. A lab, what others have done. Sometimes, if you're in a rich lab, you can, while standardizing, you can pick up 3 or 4 techniques. But okay, for the same tissue, I will have these 4 protocols or 3 protocols, and try to run these 3 or 4 samples derived from these 4 techniques on a mass spec and see which gives you the best proteome coverage, how many proteins you can identify, right? No method is 100% foolproof. Next, if you have lots of money, you can use compatible mass spec compatible detergents, but again it costs money. So Rapigest is by Waters, N-acetyl, B-glucopyranoside, azo, PPS silent, sodium lauryl, sodium deoxycholate. These are costlier, patented and sometimes patented methods by which you can digest your proteins. Protify they have lots of methods by which you can get better proteome coverage, but you have to compromise on the money part, right? But always avoid 220, Triton X, NP40 and polyethylene glycols of any kind, because not only will it be removal be a big challenge, the worst part is even if you have a very small carryover, it will destroy the mass spec. Also, you can use some additives, like if you want to work on a cell system, you don't want the cells to burst immediately, so you can use salts to mimic the physiological ionic strength. Always use protease and PTM inhibitors, right? So although we are working on ice, we are working in a cold room, we are doing it cold and fast, there would be breaks in the cold chain. A very simple break in the cold chain is you pick up the microcentrifuge, you hold it in your hands. Immediately when you're holding it in your hands, you're transferring your body heat to the tube. Sometimes when you go for tissue disruption using something like a sonicator or a rotostator homogenizer, that friction will generate heat. So cold chain is most likely to break, even if it is a very small milieu, a very small microenvironment. You want to do that, if you want to avoid that, use protease inhibitors. There are tablets like the Roche complete mini protease inhibitor cocktail. That is same you have from Thermo. You have most of the suppliers like Biorad, they would provide you. So you have to use this inhibitor cocktails. You can also add nucleases like benzonase for if you want to do some DNA, RNA binding studies, right? So this is the buffer. But when you put the tissue in the buffer, if it's a solid tissue, the tissue would not start breaking up on its own. So we use something known as tissue disruption. So the point of tissue disruption is that you have to break the tissue structure and allow the cells to flow apart, and then you have to break the cells as well. And there are multiple ways you can do it. First is the Downs homogenizer. You can see the photograph. You have a glass pestle, and with that glass pestle and a glass tube, you can plunge it. Again, when you are using these are also things that you need to standardize. If you are using a Downs, you have to standardize the number of strokes that you are using to homogenize. It might seem funny, but suppose I do 10 strokes for one sample, 15 strokes for the other sample, and I keep changing it throughout my length of PhD or throughout the length of my project, it will impact again the data that you are getting. And it will impact everything. And no amount of AI/ML can save you from a stupid researcher who has not done his baseline correct. So always remember, when you are at the wet lab, be extremely vigilant. If you are using 10 strokes, for the next 3 or 4 years, you have to use 10 strokes. You can also use instruments like bead beaters. You can use freeze thawing. You can use sonication. The purpose primarily is effective cell and tissue lysis. So you can use mechanical. So the first few were mechanical. Then you had sonic. Sonicator primarily gives you strong ultrasonic beams that breaks the tissues. Fact of the matter is that you have to break down the cell and so that the proteins can come out. There are two major categories. One is cell lysis, where we start off with cell, like you are doing mammalian cell culture. You are working with microbes like bacteria, right? You are working with exosomes. So here you don't really have to break down the tissue part. They're already freely floating cells. Next is tissue or other lysis. So you're working on plants, yeast, fungi, or harder tissues like bone. You can do proteomics of bone as well. So there you have to use some mechanical force so that the cells that are arranged in a tissue form, they are separated. So we'll focus on cell lysis methods. So denaturing buffers. So it will immediately lyse small mammalian cells and exosomes. Why? So there are, and you can also use non-denaturing methods. So if you use urea, which is a cheotropic agent, it will disrupt the plasma membrane, right? And the cells will burst. Also you can use osmotic swelling followed by shearing or sonication. So shearing force is generated when you use a Downs homogenizer. So if you take your cell line pellet and then start putting your buffer, and then you do a Downs, 10 strokes of the Downs, the shearing movement, the shear forces that are generated in the liquid, that will break. Also, you can use sonication, strong sound waves, ultrasonic waves. They will disrupt the cells. And whenever you use, always you have to remember that you have sonication generates lots of heat. And the first part we had done, that we have to, most of the samples that you process, you have to avoid heating the sample. Don't heat the sample, because if you heat the sample is gone. Your integrity of the sample is gone. So for probe sonicators, you usually keep it on ice. For water bath, you can have a cooling system. There are other kinds of bioreactor sonicators which are effective in small batches. Then you have adaptive focus acoustic sonicators, AFA sonicators. These come pretty costly, they are like 20 lakh plus. So where you can focus the exact point where you need a sonication to happen. Also, in special cases where you're using bacteria, yeast, you can use lysozyme to weaken the cell walls, but avoid excess use, because when you're using lysozyme, you're adding a protein contaminant to your system, and then this lysozyme will start doing what? It will start doing, suppose I put lots of lysozyme and then I go till peptides, What wrong did I do? Suppose you have lots of lysozymes. You have added a foreign protein into your system. Bacterial, you're working with bacterial pellet, and to that bacterial pellet you have added lots of lysozyme. What would the biggest disadvantage be when you're running it on a mass spec?
It will degrade the protein that we are working on.
What else? Thanks. We were discussing about removing abundant proteins. We don't want to add noise to our system. But when you add lots of lysozymes with the hope that it will break down all my bacterial proteins, you can add lots of lysozyme, but then you again have to have a step where maybe by affinity purification you will remove all the lysozyme that was in that homogenate. Else, it's like adding lots of noise in your own data and then complaining about it. So remember, when you are planning these sample prep techniques, the reason we are having a 2-hour lecture on sample prep is everything is contingent on your sample prep. You cannot blame the mass spec center that they give you wrong data. They will give you a strong, huge peak of lysozyme, and that peak is so high in dynamic range, the rest of your effective molecules that were actually running the game, they were masks. Right? It's just like a huge fog cover. And now your flight cannot land because it cannot see the runway. So be very cautious when designing your experiments. Always consult a few research papers, how they do it. If you're having lots of problems, talk to the scientist who has done such a work. And that's how we met with Professor Sanjeeva as well. And we were having problems. I think we have Shambhu here, who is a student who we have been mentoring since 2017. So that's how we met, and we started off doing all sorts of wrong things until we met him and he started putting things right. Of course, you have to maintain sample temperature. So key considerations if you are doing for cell lysis is maintain sample temperature. This is also true for tissue, and of course avoid balance reagent addition like unnecessary proteins like lysozyme, because that will impact your downstream methods, and I told you how it will impact, especially if you're working on a DDA instrument. You will not get signals, and even with a DIA, no instrument likes noise. Next is the soft tissue extraction. So you will have probe sonicators. Sometimes we use bead beaters. Sometimes we use liquid nitrogen grinding. It's very economical and effective. So if you have access to liquid nitrogen, nothing is as good as liquid nitrogen. Take a traditional mortar pestle, put your tissue, pour liquid nitrogen. The moment you pour liquid nitrogen, it's very brittle. Use a cryo gloves, grind it into a powder, transfer the powder into a small appender vial. Store it at -80. Your sample is good for the next 2 to 3 years. Nothing is going wrong until and unless you repeatedly freeze straw just for the fun of it. If you have a very large volume, suppose you're working on plant samples, we'll come to it. So for plant samples, a big challenge is plant samples are usually not very rich in proteins. They do have proteins, but they have other components. So to get more proteins, you have to get more of the sample. For that, you have to have a dedicated blender. Bead beaters, cryomills, cryofractionators, pressure cyclers. So there are many ways of homogenizing tissues. Again, your biology, your sample volume, your downstream methods, everything will dictate what you want to do. So this is homogenization. What you can do next is, you see, after homogenization of a large tissue, what happens is you have broken them into single cells. Most of the cells are supposed to break down by the shear forces that you have generated. Few might not. Few might not. So there you can use a probe sonication method, or you can put it in a sonication bath sonicator. Those strong ultrasonic waves will rupture the cells further, and now you have a lysate where all the cells are broken down. Sonication also helps in fact that it fragments the DNA. All the DNA is completely fragmented, and the protein solubilization, suppose you're working on histone proteins, now the protein part is completely intact, but the DNA that was bound to histone is broken down, so that helps. Then one part is centrifugation. I think this is basic biochemistry or life sciences. Anybody who has been in a lab, the one thing that we always do after homogenization is we centrifuge. Why? Because we want to remove the debris, the parts that were not centrifuged properly. So I think we can skip the summary part. Next part is, once you have done the protein extraction, the very important part is how much of proteins have you extracted. It's very important. How much of the protein did I extract? Did I extract a protein at all? Anything could go wrong. Somebody, you used a buffer that was wrongly labeled, right? Or you used a buffer which was contaminated, so it did not work well, and your extraction failed. Now you won't want to add protease just directly to the homogenate. You want to measure, and even for adding a protease, you just cannot add protease randomly. It's not. You have to measure the molar ratios, how much of protease or the enzyme would you add, and how much of substrate you would add. So these, to know how much, you have to go for extraction of for estimation the protein efficiency. So it few things it does is assesses the extraction efficiency. It will match the protein amounts for downstream application, and also it will optimize reagents, like for peptide purification, like for adding the trypsin that you need to add. So you have to really please do follow this. So extraction efficiency is, sometimes you can use a sonicator, sometimes you will use a Dunce homogenizer. Even that part you can standardize. Which part is getting more protein yield for your, so lots of standardizations to consider. And while we are using quantification, you can use two types. One is colorimetry based. So if you're working on a highly abundant protein, you have no dearth of samples, you can use colorimetry based methods. If your samples are very rare, you're working on a clinical sample, right? And maybe it's a very rare cancer, or you're working on a sample type that is archival. You're working on FFPE, right? Tissues that has been put in paraffin and then it has been sectioned, so you don't have much of a tissue to work with, and you cannot waste too much of your protein just in estimating how much of protein you have, right? Then you can use very sensitive methods that are fluorescence based. But always remember, the method you're using to estimate your protein should be compatible with the buffer that you use for extraction, one, and number two, your buffer should not have any inhibitors for that specific protein quantification methods. Each method will have a set of inhibitors. Now if you start using the incompatible methods, you get wrong results. So you have the BCA method, and it reduces the peptide bonds from Cu2 plus to Cu plus, and you can get the absorbance at 562. You have the Bradford, you have the Fallin-Lowry method. Alternatively, you can also use a UV visible spectrophotometer. If you have a microplate reader or a nanodrop, you can use it at 280 nanometers. But remember, it only looks for aromatic amino acids. So every technique would have its flaws and pros and cons. So, or else you can use fluorescence-based methods, but they are much more sensitive than colorimetric methods. So you can work, if you're using with a low abundant protein, of your sample is very low abundance, you don't have too much of your sample to spare, you can use intrinsic tryptophan fluorescence, or you can use reagents like the orange assay. So they use a specific dye and it is excited at 485 nanometers and it has an emission of 590 nanometers. And you can use it with fluorometers, microplate readers. So there is one reagent that is called the CBQ-CA, a sensitive fluorogenic reagent. So it detects amines in the proteins and it has a very good dynamic range. It has give a reliable for low abundance protein quantification. So see, if you have lots of proteins, usually it's not a problem. It's only when those people start working with low abundance proteins, or the sample is of very low abundance, you talk to your histopathologist and the pathologist says that I will give you 10 sections, 10 7 micrometer sections of the archival tissue to do your proteomics with. That does not hold much of protein. So you have to extract those proteins and you cannot waste much of the protein just to doing the assays and standardizing stuff. So how do you choose it? Colorimetry, cost effective, uses common equipment, best for mid-range protein concentrations. Fluorescence, highly sensitive, accurate, ideal for low protein concentrations and complex samples. And always consider the buffer compatibility factor. Do not forget that part. So I will skip. So this is what a Bradford assay looks like. This is what we used to do at proteomics lab. So you do it on a microplate, and I think most of you would have done a Bradford assay, but that has nothing to do with proteomics as such. Next, we will move on to a section that is called proteolysis. So most of the things that we are doing here is bottom-up proteomics. I think we have discussed this in detail multiple times that we are doing bottom-up proteomics. And in bottom-up proteomics, we have to first break down the protein and then we will recreate the picture of the protein at the last stage. Other one is top-down. We are not discussing that. So what does proteolysis do is it hydrolyzes proteins into peptides, and these peptides can fly and these peptides can be recognized by the mass spec. So optimal peptide length for mass spec is usually 7 to 35 amino acids. And one of the challenges, if it is a very long peptide, it is very difficult to identify and it might bind irreversibly to solid phase extraction methods that we will use sometime later. We'll just learn. If it is very short, just like those who are familiar with primer design, if your primer is very short, the DNA primer is very short, it loses specificity. If your peptide is very short, that peptide could be any peptide. Now 7 amino acids, that means almost 21 nucleotides in codons. That gives you a good amount of specificity that your instrument or your software can probably identify better, right? Short peptides, they lack specificity and it will only add ambiguity to your data. So we want 7 to 35 amino acids. Longers we cannot accept, shorter we cannot work with. And here, although we use the term proteolysis as trypsin digestion, so digestion is a very general term. In scientific literature, avoid using the term digestion. You can use trypsin proteolysis because proteolysis is hydrolysis of peptide bonds, and that is what trypsin does, right? Digestion is just breaking down. It does not involve the specificity of an enzyme catalyzed reaction. So whenever you use, although in common terms, maybe in your lab, we'll just call it as trypsin digest or tryptic digest. Next is why trypsin. So trypsin is considered the gold standard. It has a very high specificity, it has a very high efficiency, the availability is there, and of course low cost. And what does it do is, it's at the C-terminal of R or arginine and lysine, they except when followed by K. So it has a very specific site that it will cut. So it generates peptides of ideal length for chromatographic separation and MS analysis, appropriate hydrophobicity for efficient database identification. You might not understand what is that, but you will surely understand at the end of day 5. Produces short peptides around, sometimes, it's not always true, depending on the RK junctions that you have, sometimes it does produce less than 6 amino acids. That limits the proteome coverage. Sometimes there are regions that are RK devoid and it causes longer peptides. So trypsin is gold, but it's not 24 karat gold. Maybe you can put it as 18 karat gold. So it has some drawbacks, but since the drawbacks far outweigh the advantages, primary advantages is efficiency, availability, and also the low cost. But what if your experiment does not work with trypsin? So you have some alternative proteases. We have ASPN, chymotrypsin, arginine C, lyse N, pepsin, proteinase K. So you also have some specialty. So it's not that you have only one way to work with. There are multiple ways and methods. But you see, if you go anything beyond trypsin, costs will be one big factor. And as I said, since you will be using this for the next 3-4 years, when you're a PhD, or if you're even doing it for one experiment, your lab would be preferring only one method. People usually stick to trypsin. If trypsin fails miserably, they go for alternative proteases. But we do have alternative proteases. And also the alternative proteases comes with issues like fragmentation. So peptides may have inconsistent charges that might lead to fragmentation. So when we get deeper into mass spec methods, you will see that once the peptides are ionized and they fly into the mass spec, they are then fragmented into Y ions and B ions. So this fragmentation is an issue with alternative peptides, because then peptides do not carry proper charges.
Making is, in many situations, and the software will show you, all of these have been implemented. So while it's useful to have a rough idea of the theory, you don't have to be able to have the exact technical detail to implement one from scratch. So knowing about what these different techniques are and how they work is useful, but you don't have to have a comprehensive knowledge in order to use them. It's a bit like, as I say, if you drive a car, there's the skill in the driving of the car, but you don't have to build your car before you drive it. So it's knowing the techniques, but what I'm going to show you is ways to start using them as well, so that they can empower you to start using those things and start discovering new things. So does anybody have any questions? Has anything occurred to anybody overnight? Is there anything you want clarifying? And this is an opportunity for you to do that. I can go over things again, explain things again. Please ask questions.
I'm new in this AI thing. So if I have a data set and I'm not aware of much of its features, like I have a very limited set, but I want to extract some more new features which are unknown to me. So is it possible to do?
This is causing an issue with the sound. You can just
Okay, yeah. So you want to find out, you've got a data set and you want to find out new features of importance about that data set. Yeah, so, but you have, it's a high dimensional data set or it has a large number of variables, you just don't know how they relate to the thing of importance. Yeah, so I can show you some more of that, and I will show some more of that coming up. But essentially the method we use is we look at each feature on its own in the first instance across the data set. We look at its ability to model an answer to a question. So say you have disease healthy and you've got all these variables. We test variable 1 against disease healthy, see how good it is at predicting it. We then test variable 2, see how good it is to predict it. Test variable 3. So in this way we're testing out every single variable in the list, and we can rank them and see which are the best ones to use. And that way we can start to see at a first pass what is important. What we can also do though is we can start building in a stepwise fashion models. So we could say, right, what's the best single variable? Okay, we find the best single variable, we keep that. We then add remaining variables to that to find the best pair of variables. So
Like this, like that, we, by selecting variables which are like significant according to our data, we can add it on in our model based on that.
yeah, so we're adding things into the model to build the model up in a stepwise fashion based upon performance. So we start off with find the best single one, then the best pair, the best triplet, best quadruplet, and so on. And keep growing the model until we stop getting an improvement in performance. So that's another way of building up a model from these data sets. Some of the methods I showed you yesterday around weights and things like that, they have limitations for these big data sets, so I wouldn't use those. But that was just to illustrate historical stuff. Any other questions? No.
So could you please explain that activation function part?
Sure. So activation function is a function which is the mathematical basis of the neural network. So this piece here is the activation function. So what it is, it's once we have the summed values, so we have this value multiplied by the weight, plus this value multiplied by the weight, plus this value multiplied by the weight, and then we sum that value. We'll get a value from that. And then we apply it to the activation function. So what that does is, okay, let's have a look at our value of the sum of the weights across here. Okay, so it's quite high. So just reading effectively off the graph. So the amount of activation is, if it's high, it goes out as high. Okay. So the number that comes from this sum is passed on to this function. So if, for example, it's very low, then the output's low. If it's high, the output's high. So it's transforming that number onto an S-shaped curve. And that dictates what comes out of that node into the next layer. So if we look at these activation functions, here, if the value's low, the value's low, and it would just linearly relate. So that's a linear activation function. That has the potential to go to infinity and the potential to go to minus infinity. If we look at a sigmoid, it tends to 0, it tends to 1. So it's dampening down those high ends, which is good, because sometimes the model will run out of control. So if it's low, let's say it's minus 6, we get a value of 0. If it's minus 2, we get a value of about 0.1. If it's plus 4, we get a value of 0.99. If it's plus 6, we get a value of 1. So the number comes in, the activation function, that value goes out. Yeah? So where it starts to have an impact as well are with rectified linear units. So this is about a decision-making process. So here, let's say we have a value of -5 coming out of that hidden node. Nothing happens, we just get 0. And they get 0 at -4, 0 at -3, 0 at -2, 0 at -1, 0 at 0. And then only when it's positive does it start to go up. So that's immediately squashing that distribution to get rid of all the negatives. So what we're doing then is choosing these activation functions based upon how they modulate what's coming into them from the network. And then by using different functions, we can start to represent different characteristics of the patterns in the data. Yeah, there's nothing stopping us having multiple hidden layers as well. So we could have a hidden layer which is full of sigmoids, then have a hidden layer full of rows. That would have a particular effect on the network, but we could try that out if we wanted to see. There's no right or wrong answer. It's we don't normally say, oh, this is this sort of data. We're going to use 3 layers of a relo and 1 layer of a sigmoid. It's a trial and error approach as to which ones we use. What we tend to find though is TANH and sigmoid work better for biological systems because there's more of that noise. It's not such a crisp cut-off as in physics systems, which would be more useful for ReLU or in some cases linear. So it's just really how the numbers are transformed. Any other questions? I'm presuming we can start. I don't know if it's streaming. I'll carry on anyway. So the next method is an alternative machine learning method. It's in the public domain. It's been used quite a lot. And that's support vector machines. A support vector machine is a supervised approach, so we're telling it the ground truth. So is it this class or that class? We can generally only cope with 2 classes. And it doesn't make support vector machines will not make a continuous regression type analysis. So what it's trying to do is find that decision boundary. So if you remember yesterday I talked about decision boundaries. We have a cloud of data. The decision boundary is the thing that separates one group from the other through multidimensional space. So this technique is reliant on a kernel. A kernel is kind of a seed function. So it's a type of function class. So kernels are a weighted function which maps the decision boundary. So it's a bit like how I showed yesterday where the neural network maps the decision boundary by putting 3 sigmoids together. A kernel will use a function that does a similar sort of thing. So a support vector machine will use a kernel that does a similar sort of thing. And we can choose what type of function. So sometimes it might be a linear function, so that's where there's a straight line decision boundary between the groups. We can use a sigmoid, we can use a log, or we can use a radial basis function. So radial basis functions are quite interesting. They're contracting and expanding spheres. So what that's trying to do is creating a sphere or a couple of spheres which are trying to define the classes. And it's the radial basis, so the radial is effectively the width, or half the width of the circle, half the diameter of the circle. And what happens is the SVM changes the weights of the function, effectively scaling the data through the multiple dimensions to maximize the distance between the points and the decision boundary. So it knows the distance from class A to the line, it knows the distance from class B to the line, and then it moves the line such that the distance between A and the line and B and the line is as big as possible. So what does that look like? Well, it's back to this just 2D plot of the population. What we have effectively is healthy, we have disease, and what we're trying to do is separate out one from the other with this line. So the first thing we do is we map the distance between each point in the population and the line. So we know the red lines have so much distance away from the line. These will be negatively weighted because they're on the wrong side of the line. And then we have the greens which have so much distance from the line. So we're mapping out that data space and the line's position relative to it. So what then happens is the line moves and that's recalculated. The line moves, it's recalculated. So the line is moving through the space to make sure that the sum of those values is as high as possible and the sum of those values is as high as possible. So in that way, by compromising, the decision boundary is formed. So what in effect we're doing is we have this line and we're moving the line, we're changing the gradient term until we find the best fit. And it's that point where we have the classifier. Oh, thank you. So that's how a support vector machine works. This is a very simple 2D plot. These can work in very high numbers of dimensions. When they were used for mass spec data, and they were used a lot around between sort of 2000 and 2010. They were used a lot because they didn't suffer from the curse of dimensionality. They took ages to train though, that was one of the problems, and even then they were limited by what they would produce. They were only really good for classifiers and there was a limited number of kernels you could use. So that's that method. Next method is decision trees. So this is whereas the SVM and the neural network are based upon a mathematical function. Decision trees are essentially based on logic. So if this, then that, or if this, then that. Now, you can use them in a regression form. Sometimes they're called CART, or classification and regression trees. And that becomes a bit more probabilistic. I'm not going to go into it here. But I'll keep to the simple form. So a decision tree is effectively a set of nested rules. Now, we look at an individual in the population, and look at the population and say, can we split that population with a rule into one set of individuals and another set of individuals? Okay? So the first rule will split into probably a pure class and a mixed class. It's very unlikely with a single rule we'll be able to split the whole population. And that would be like effectively two groups with a massive distance between them. It would literally be, they are here, those are there. Reality of biology is more complicated than that. So each rule splits the data into a pure class and a mixed class. And then further rules refine that splitting. So again, we're nesting and building multiple rules in in order to classify as many individuals as possible. So the decision boundaries and the rules will give a separation in each dimension. So if we have a large number of dimensions, we've got lots of potential to generate lots of rules. So it will look through and see which ones are the best ones. So it will scan through and find the best rule in this variable, or the best rule in this variable, and it might see, well, on its own, this variable with this rule, does the best. And then it'll take that and then nest the next rule off of that. So, and then it constructs a tree which represents the overall model, which is a whole load of rules that are nested. So, if we take this example, we have our first rule, and we're taking that on the x-axis. So what you can see that the line is moving and defining a group which is a pure class and a mixed class. Right. So our first rule is saying if less than x1, because it's on the x-axis, then it's a pure class, or if greater than x1, it's a mixed class of healthy and cancer. So you can see that's the logic of it. It's an if-then rule. So there's a threshold there. So what that might look like in our first branch of our decision tree is if less than x1, it's this group, and if it's greater than x1, it's that group. So we're starting to classify the population based on rules. So what we do next is add a further rule. So we have our x2 rule here, and we can say now, if greater than x2, it's a pure cancer class. If less than x2, it's a mixed class. You see, so now with our next rule, we've explained a different proportion of the population, a different part, that effectively wasn't explained by the first rule. So that would look like this. So we take the x1 group, we split that into a pure cancer class, and we split it into a mixed class. Okay? And then we have our third rule, and now we're looking at the y-axis, and we're now saying, and it's the feet of these people that we take as the point where they, in the space where they exist. So we're now saying if less than y1, then it's a cancer class, and if greater than y1, it's a healthy class. So with three nested rules, we've explained that whole population. So that's using a logical structure in the tree. But broadly, if you look at this, you can see we have got effectively a decision boundary, which is sort of a little bit sigmoidal in its nature. So now we have a classifier. We can say, well, we put our cases through this decision tree and they get filtered out into particular groups. And what that looks like is something like this. So we take x1, our rule there, take x2, our rule there, take y1, our rule there, and we have a way of classifying into the two types. The problem is, like everything else, this now is probably overfitted. It's a small population, it won't generalize well. So in the same way with a neural network, we have effectively mixed up the data into different groups. We can do this with a decision tree as well. So what we do is we take lots of trees, and what do we have when we have lots of trees? We have a forest. So a random forest is taking lots of splits of the data and then creating a tree.
This is the main body of the shell of the shirt, right? This is the collar. If you remove those marks, even the best tailors cannot stitch it back. So when your protein degrades, you are not keeping any mark on the protein. When you're using a specific enzyme like lysine, which is the mostly used, you're keeping a very specific mark that I will cut only at those junctions where trypsin cuts, right, between lysine and arginine. So that breakpoint becomes your identifying mark, and you know this has been cut by me, not by degradation. And this is very important when we go back and try to trace back via bioinformatics tools like MaxQuant, MS Fragger. There are multiple tools by which you can use the raw MS data and make sense of it, that which are the proteins that we have identified. That is very important. We will come to it. Then we have to desalt the peptides. The reason we have to desalt is we have used lots of buffers, and we will discuss about lots of buffers. And the mass spec is a very sensitive instrument. We cannot just put anything inside it, right? Professor Srivastava was telling that we have to use a very high voltage and electrospray ionization. He was talking about John Fenn, and who had developed this technique, because primarily mass spec has been around for almost like more than 50 years. But biologists started using it only after John Fenn told them that yes, you can ionize peptides. And he's developed electrospray ionization. So for electrospray ionization, if you have salts, it will start acting as inhibitors. It will stop the ionization. And you see, the better a peptide can fly, the better it will be identified. Right? You know all celebrities in India, right? Name one celebrity. MS Dhoni, Virat Kohli, Sachin Tendulkar. So they are high flyers. We can identify them very well. But do you know who plays for Ranji Trophy in Gujarat, or maybe Uttar Pradesh? Probably not. Do you know all the uncapped players of IPL? Probably not, because they're not high flying as yet. So mass spec is just like us. Only those who can fly well, they will identify. And if you put lots of salts, they will stop flying. So desalting is a process that removes the salts, cleans the peptides, and these peptides can then fly into the mass spec by electrospray ionization. Beyond that, we have this instrument that comes in many shapes and forms, multiple vendors. We have Thermoscientific, we have SciEx, we have Bruker, and these they can always make newer technologies. So Bruker has timsTOF Pro, timsTOF Pro Flex. Thermo has come up with Astral. It has Orbitraps. So multiple kinds of, you have Agilent, who makes primarily TOFs or time of flight based instruments. So instrument can be various, but sample prep techniques are identical for most. I think I can skip this. Right. So what do we need to consider when we are preparing the sample? So you have to always, before you start doing an experiment, please do a mind experiment as well. That what is your ultimate goal? Are you trying to identify phosphoproteomics? I mean, you want to do phosphoproteomics, right? Now for that, your sample prep technique would be different. You have to do some phospho enrichment using IMAC columns, right? You want to do glycosylation studies or any type of post-translational modification studies. Your first impact will be your sample prep technique will change. Next, what is your long-term project goal? How much reproducible do you want to be? Are you handling low abundance samples? Now suppose you are working with a multi-center project, right? Now very recently, it's a very recent thing, there is a group at IISc. I think Professor Sanjeeva was talking yesterday, I don't know if he has talked, but his lab is starting this project on longevity, where they are trying to sequence the proteome of aging. They want to know what is normative aging as we age without any diseases. How does your body proteome change? So they are doing a multi-omics analysis. proteome, metabolome, metagenome. Now it will be a multi-center study. Multiple labs will be involved. So my long-term project goal is reproducibility across labs. So you can't use instruments or you cannot use protocols that are very sensitive to environmental applications. And these are real-world experiences that we have. Few experiments can be done very easily at Allahabad. Few experiments can be done very easily at Kolkata or Mumbai, just because the weather is as such, right? How would you carry your samples? Are you taking up samples from rural villages? So you have to plan it ahead. Before you hit the test tube and micropipettes, you have to plan it all ahead, and that's where experience kicks in. That's why senior PIs can tell you that, okay, don't do this, don't do that. Handling low abundance. Sometimes, like for the COVID work that we did at IIT Bombay, the COVID virus, we wanted to detect it on the viral transport media, VTM. Now VTM is 5 ml, 6 ml. They push a swab into your nose, getting here, right, in the upper palate. How many viruses does it catch? Maybe a few million, but viruses are very small. So most of the VTM is primarily the VTM. I mean, it has lots of bovine serum albumin, and even your cells from your own body have now been part of the VTM. To find out the viral protein from there, you have to do some sort of enrichment. So if you are going for a low abundance protein, likewise I did not tell, but sometimes you are working with highly abundant proteins, plasma samples. You want to check what is the difference in expression of a certain set of proteins. Most instruments, most mass specs, right, will get overwhelmed because it is getting some very strong signal, maybe from the immunoglobulin part, right, from the globins, so from the albumin that is there in our blood. So how do we remove that? Any instrument, even us, we have a way of separating out. Imagine yourself sitting in a local train. It is rattling through the track. Have you ever felt that you have to shout much harder to be heard? Because there is lots of noise around. Now these highly abundant proteins will create lots of noise in the mass spec, and if your instrument is running on a DDA mode, that is data dependent acquisition mode, it will lose out all on the sensitive samples. And you see, when we are talking of a systems approach, even the smallest perturbation can have the largest impact. So then again, you have to design your experiment in a way that your highly abundant proteins do not mask the signals, and most signaling molecules like cytokines, even the small signaling receptors, they are usually very low amount. They are not highly abundant, and if it is masked in the mass spec, then you lose out. So people who are doing cell surface proteomics, there are people who are doing, so there are ways of enrichment. I won't be able to tell you everything that I'm giving you the fundamentals, so that while you plan your experiments, you do it likewise. And of course, you want to do global proteomics. So suppose you want to check all the proteins in the brain, all the proteins in the liver, and all the proteins means membrane proteins, it means cytosolic proteins, it also means mitochondrial proteins. Or you want to check only for the nuclear proteins. Now for a cell, if you want to do a nuclear protein, the best easy way will be to go for a nuclear fraction. If you want to check for mitochondrial proteins, an easy way would be to go for the mitochondrial fraction. So here, your knowledge of biology, your knowledge of analytical tools like centrifugation comes into play, right? You want to do cell membrane proteomics, then you have to do something else. You want to just do the cell surface, you want to go for protein-protein interaction, again, separate set of experimental design. And ultimately, the bottom line is, your choice of protocol will define the results you get. But then you cannot blame us, or especially Professor Graham, that he taught us wrong, and see, all the results we got is not what we were expecting, because you started off wrong. So the data analysis part comes much later. This is something that you can do at your own lab most of the protocols for protein extraction and peptide synthesis, making till the peptide part, can be done in any lab, anywhere. Even if you're a very poor lab, you can do it. For mass spec, you need big instruments, well-funded labs, but you can always outsource that part. So these are a few books that you can study. I cannot share books with you, but these are the names that you can use. Also, I will share with you, if somebody reminds me at the end of the course, a few papers that are very good, are very wholesome in ways of by which they have taken up proteomics. Those are, you can see there is a review article almost 80 pages long. I could share it right now, but it won't make much sense. So I will only give it to you when it starts making sense to you. Right now, 80 pages, you would get overwhelmed. So, and of course, there are always few studies that some people have done earlier. So if you're having any protocol, you want to do proteomics of tomatoes, look up on the PubMed, look up on Google Scholar, get in touch with that scientist if you can, ask them for help. 9 out of 10 people would help you. The good guys do. Else they might have retired or something. So also one thing is the protein extraction. So first part is how do you extract protein, right? Protein is in the cell, right? You have to extract it, you have to take it out, and not only do you have to take it out, you have to sometimes fight with other molecules that are interacting. Suppose you want to take out histone proteins. You want to work on histone proteins. You have to fight with the DNA so that the DNA leaves the histones, and then you have to take it out. So it is highly sample dependent, the type of sample you are dealing with, and it has also, you have to align it with your downstream assay requirements and experimental goals. So nothing, as I said early in the morning, it's not my neighbor lab is doing this, so I can do this. Might be you would get all the wrong results if you do that. So what we want to do here is learn the mechanisms that help us in preparing the samples and some efficient tools, and mostly these are for bottoms-up proteomics, which means we start with a broken protein and we try to identify the entire proteome from that. There is a top-down approach as well, but we are not going into it. Okay, so first thing is your choice of buffer. What buffer would you use? Mostly it's 8 molar urea in 100 millimolar tris, pH 8.5. pH 8.5 is for trypsin activity. Again, these are very generic methods that most labs follow. If you have anything specific, it might not suit you. But suppose you're working on a soft tissue or like brain or liver or kidney of any animal, you can use these buffers. Then you have to use desalting methods like stage tips or set pack columns to get clean peptides. Always avoid some detergents like Triton X100 and NP40 because they will always impact your downstream analysis. So remember, it's not only about what you need, it's also about what you should not. Learn the do-nots first. The do's can be taken care of later. So just to recap, so your study goal and sample type, that determines what kind of buffers you will use with. Extraction conditions can affect results. One rule of proteomics is cold and fast, right? Most of the samples you have to process in the cold. So if you have a cold room, very good. Go ahead, use the cold room. Wear a jacket. And it's very pleasant in India if you are working on proteomics. It's in the heat of June. You can walk into a cold room, it's 4 degrees Celsius, stay there for a couple of hours. You can have a hill station experience. Again, throughout your experiment, once you have started, especially this is true for PhD students, it's 4 to 5 years journey, right? You have started with a protocol. You have optimized it 1.5 years. After 6 months, you suddenly find you have generated data for like 2-3 experiments, 4-5 experiments are still pending. Suddenly you find there's one very good way of getting proteins. Be very careful. You cannot use an apple protocol for one and an orange protocol for the other set of samples. If you have started with an apple, even if you get a better apple or a better orange, don't. So old is gold. Once whatever you have standardized, stick to it. Your lab might move on to another protocol. Maybe your young friend who is starting off with the PhD, they can move on to a newer protocol. You should not. And that's true even for softwares like MaxQuant that we will be discussing. So throughout your analysis, use only one version of the MaxQuant. Don't change versions, because these are algorithms, they keep on changing, and that might heavily impact your data analysis. So stick to conditions. Once optimized, stick to it until the end of your project. For your PhD is like almost 3 years to 4 years that you generate data. Stick to it. And that will help you with reproducibility, and consistency is the key. So getting into global proteomics, a good buffer that should have, as I said, 7.5 to 8.5 pH, and you have to combine with strong denaturants for effective. So what is a denaturant? Why do we need a denaturant? Why? So primarily the cell is a very robust, it's very robust, right? All the proteins are there in its own place. They won't want to break off, right? Since they don't want to break off, you have to use some agents. We also call it a chaotropic agent, so that induces a lot of chaos, and the proteins will break down. Another good use is most of the proteins, most of the cells carries endogenous proteases, right? And these endogenous proteases can start degrading a protein even before you know it, right? You said, I did not add any trypsin, I was working in the cold, but enzymes have an optimum temperature of 37, but that does not mean at 4 or 5 degrees they will stop working. They will work with very low efficiency. So denaturants, on the other hand, they will denature the protein, it will break its native shape, and once an enzyme loses its native conformation, it stops acting. One consider that you need to always consider is cost. In any lab you go, because you will be doing this experiment for the next 4 to 5 years, funding might not be as smooth as it is today. Tomorrow you might run out of money, so don't go for anything that is very exorbitant and that you cannot continue for the long run. Next, volatility. So most of the buffers that we use, like ammonium bicarbonate, are volatile buffers. You can just sublimate it, right? A good way of getting rid of a buffer is to just dry it on a speed vac, and it will just sublimate. So get rid of the buffer easily. Reactivity. So primary amines that are present in many of the buffers might react with your amino acid groups. And since we will be identifying proteins based on your amino acid composition, that's what a mass spec does. Mass spec will weigh your amino acid and then tell you that, okay, so it will weigh something and then it will tell you, since it is this much weight, this is the charge, this is the mass. Based on this mass by charge ratio, according to my database, this could be valine. Now this valine gets amidated at one point, there's a high probability it will misidentify. And if it misidentifies one amino acid, most likely by a chain reaction, it will misidentify the entire protein. You don't want to do that, right? So we'll just go through the few buffers. Tris buffer, low cost but non-volatile, contains primary amines, may chelate metals, and may not be compatible with labeling methods like TMT labeling. So I know you might not know right now, but there are techniques such as TMT labeling that can there are methods by which you can do proteomics. One is the labeling method, and sometimes TMT labeling won't work. So again, always try to remember what is the downstream method that you are going to follow. Are you going to label your proteins? Read up. Next, ammonium bicarbonate, ABC buffer. Most labs will call it ABC. So it is very volatile. It can be removed by lyophilization, which is put it in a speed vacuum, dry it off, but again it might promote methionine oxidation. You have HEPIS buffer. It's costly, so that is again a problem. So this is a table where we have multiple buffers that you can use for protein extraction, and there are a few notes which tells us when not to use the fan, when not to use something and when to use something. I would request, whenever you plan a proteomics experiment, always go through the entire experimental procedure in your head before you start hitting the wet lab. Also, you can use always online tools so that your calculations are correct, right? Don't rely too much on seniors' notes. Always try to rely on good books, and if you have to scale down something, if you are very good at maths, do it, but then get it verified by somebody else. Else there are lots of online tools available where you can do the calculations, but fact of the matter is, if you go wrong even by 1 millimolar, your batches of buffers will be different, and your results will show that there will be batch effects. And always remember that all buffers do not work with every method or equipment. So the downstream should be kept in mind. I think I have just discussed this part, why you need to denatures. Why? Because it alters the endogenous enzymes like proteases and kinases. Suppose you are wanting to work on phosphoproteomics, and while your extraction you have triggered a kinase, then you don't know, the exact results. You might still be able to do phosphoproteomics, but it would not be a reflection of what you want to see, right? It would be a reflection of how you handle your sample. You manhandle your sample, and your new sample, the way you handle the sample, the results will be like that. Denatures and surfactants are there to solubilize proteins effectively. So you will use surfactants or detergents that you can solubilize the protein. How do you choose it? Again, multiple ways. First, must work well with your protease, because ultimately you would need a protease like trypsin to digest your proteins, and before you are not removing the salts before the trypsin digestion. So remember, you cannot use a denaturant that will denature trypsin. Trypsin is also an enzyme, it's a protein, so you cannot use a denaturant that will still be in solution and start impacting trypsin activity. Also consider how your denaturant will affect peptide purification, because again you will be using a setback column to purify the proteins, the peptides. You have to remember that as well, that how will this cleanup process get impacted. Why do we, the most common denaturant that we use, as we discussed, was 8 molar urea. And why we use that? Because it is compatible with trypsin if you bring down its molarity to 2 molar, right? We started off with 8 molar, then what we do is we add lots of ABC buffer and we bring down its molarity to 2 molar. Now 2 molar urea will allow trypsin to act. That is what you can do. You can dilute the denaturant. At no point can you take off the denaturant. That is an impossibility. Next, it causes carbamylation when heated. So, as I said, much of the work that you will be doing will be a box of ice or in the cold room, because heating impacts number one, the protein starts to degrade or hydrolyze, and it will create a smear pattern. And second, it will cause carbamylation if you have urea along with you. And urea has one more good thing: you can always remove urea by desalting techniques. Then you have a very famous denaturant, sodium dodecyl sulfate, SDS. The good part of SDS is it's much better when it comes to extracting proteins. The bad part of the story is it's incompatible with trypsin and reverse phase chromatography. Because LCMS, LC stands for liquid chromatography, and it's usually reverse phase chromatography. So you can use SDS under special conditions, but then there are special tools and methods by which you have to remove SDS before you go for trypsin digestion. And when I always take there are special tools and special methods, it adds to your cost of doing the experiment. And when we are talking of big data, you always remember you have to process not one but maybe 20 or 25, or sometimes if you are working with a big project in humans, maybe 1,000, 1,500 samples. There's a very good paper by Professor Hammerstein from Harvard, where he has processed around 1,500 or more than that of human plasma samples. And even sitting at Harvard, he did not have the money to use depletion columns, because each column costs around 14,000 rupees in India. But even at Harvard, in US, they don't have that kind of insane money. So always remember, doing science is one thing. The smarter thing is doing science with the money that is available with you, with the techniques with the expertise that is available with you. Don't have very grand plans. It will lead to frustration. So again, choosing the right denaturant. So these are the different types of denaturants that you can use. 8 molar urea, the best thing. It's non-volatile, it's chemically reactive, limited heating, but must be diluted less than 2 molar before trypsin addition. You have 1 to 5% dodecyl SDS. It's cheap. It's a very strong denaturant, but must be removed before trypsin digestion. You have another 1 to 5% sodium deoxycholate. Is hydrophobic for membrane proteins, easy to remove due to precipitation with acid. Another is N-dodecyl B-maltoside. It is expensive. Low amounts can be used with trypsin and LCMS. So then Tritonix 100, do not use it. If samples are already having this or NP40, you have to again go for protein precipitation to remove Tritonix. Tritonix, NP40, polyethylene glycol. There are a few chemicals that are mass spec killers. They are serial murderers. Even a small bit of it enters the mass spec, your machine is gone. You have to call the engineers. And the best practice is prefer tris buffer over ammonium bicarbonate if you want to minimize oxidation. Method compatibility for intact protein separations, that you want to do intact protein by size, choose denaturants like SDS, and again you have to choose whether those methods are compatible, and always have this in mind that you have to go for workflow integration. So anything that goes downstream should be compatible with what is going on the upstream, right? Again, and one size fits all solutions are not available. So there are some methods by which you can remove the detergents. One is 1% SDC or 1 molar urea can be used with trypsin lysis proteolysis, and you can achieve proteolysis compatible concentration by diluting with appropriate buffers. Like I said, for urea we use 100 millimolar ABC or ammonium bicarbonate at pH 8.5. Does two things: reduces the levels of urea to 2 molar. Second, keeps the pH 8.5, which is extremely important when you have to digest with trypsin. Then if you have to go for precipitation, you can use acetone, trichloroacetic acid, chloroform methanol, water. You can use solid phase method like S-trap, which is something that is patented by Protify. You have filter aided separation techniques like FASP, and you are also having some specialty detergent removal columns by Pierce, Thermo Fisher. But all these methods will do two things: add costs and time, and also I would add one more point. it also reduces the amount of protein you have to work with. See, we tell that all this is a standard